to the final keynote of the second day of the 20th ISMB. 3 years ago, ISCB started its Fellows program, and last year we have begun the new tradition, which is to have the Fellow of that year give a keynote. It's my special honor to welcome today Richard Durbin as the Fellows keynote speaker. Richard currently is the joint head of human genetics at Wellcome the Sanger Institute in Hingston, close to Cambridge. He did his PhD and his bachelor in mathematics, his PhD in '82 in biology in Cambridge, did a postdoc in Stanford, came to the MRC in Cambridge, and since 1992 he took on the challenge of heading computational biology in Sanger. Now, Richard has one of those outstanding things in his career that I don't see very often. He was participating in the International Mathematical Olympiad. And for me, I have always wondered when I see these outstanding geniuses, what happens to them? What a pity that they don't come to computational biology. This one, we as a field recruited, or he recruited us, or whatever way you look at it. Richard has an ISI Hirsch index of 87. Picture that. The top paper quoted almost 10,000 times, 6 papers over 1,000 times, 75 over 100 times. Now, he's known for things such as ISTB, worm-based, GeneWise, started PFAM, Ensemble, TreeFAM, and had his finger also on Hammer. Had his finger in the development or in the use of hidden Markov models overall for sequence database comparisons. Was co-leading the 1000 Genome Project and now the UK 10K collaboration. Incredibly exciting data, a mixture of experimental and computational biology. Richard, I'm very happy that you are here with us.
Well, thank you, Burkhard, for the introduction, and thank you to the Society for electing me a fellow. It's a real honor, and there are a lot of very impressive people there and here. And thank you for giving me the opportunity to talk to you today. So I want to talk about doing genetics, essentially, with the wonderful new sequencing technologies that we now have available. And it's always good to start, I think, by realizing, I find the most striking way to recognize the influence of genetics on human biology is to look at identical twins. These are people who have the same genome, and we know that they are much more similar than another pair of individuals who have differing genomes. Obviously, there are differences, they're individuals, and there's a contribution of the environment. But it's a sort of direct, natural experiment to show the contribution of the genome, and we know there's a strong genetic component to human variation. So there's two components to genetics, really. One is to find the genetic variants, the variants in the genome, and the other is to look at variants in other traits of interest in biology, and to try and join them up, connect those together. And I'm going to aim to talk about both aspects in this presentation. But something that genetics has been an important subject for 100 years and has contributed to biology for a long time, but it's actually only in the last few years that we've been able to look directly at the genome sequence. And the direct way to study genetic variation is obviously to sequence genomes. And as we all know, that's become easy, comparatively easy, very rapidly. So the first genome cost $1 billion only 12 years ago. Just 3 years ago, there were of the order of 200 human genomes sequenced, and the cost per genome was about $50,000. This year, there are thousands of genomes available for research or being sequenced. And I think, more or less, the cost, you can get a human genome sequence for about $5,000, another factor of 10. People talk about the $1,000 genome. I think that is coming, at least in terms of the raw sequence. certainly within the next few years, and they will be obtained for research interests, for personal interest, and clinical usage. So the use of genetic information will extend well beyond research. But there are a lot of research opportunities that will come. And I think it's not unrealistic to expect, looking at this sort of exponential growth and the value, that there will be millions of human genome sequences within the next decade. So the growth is much faster than Moore's law. It stretches computer science. And a lot of people are concerned that the costs of analyzing the data may outstrip and totally dominate the costs of generating it. I'm actually more sanguine. I think it's a question of scaling. It's you in the audience, actually, who are working out how to do this efficiently, and I think there's scope for keeping the computational component efficient, but it's certainly challenging. So we're all related by a complex structure of inheritance. There are segments of DNA which we share in patterns between individuals. They're separated by recombination events. And there are differences caused by mutations since the common ancestor. And so a key question from my point of view is, we have a reference genome, but does that really represent, is that a good basis for understanding the sequences of all the people in the audience? And I think really we want to build a richer representation of the human genome as the genome of the human species, essentially all of our genomes, how they're related to each other, so that we can place any one individual into that. And that will provide optimal information and efficiency for functional genetics. And it's also the foundation for human history, studying human history and evolution on a genetic basis. So as the sequencing technologies became available, and it became clear that it would be possible to do this, a public consortium was put together, the 1,000 Genomes Project, to basically take the first steps in this direction. And that was in 2008. The project was started. There are multiple centers across the world. Multiple platforms were used. Multiple funders, NIH, the Wellcome Trust, the German Max Planck Institute, the Chinese Beijing Genome Institute. And the aim was to sequence a large number of human genomes so as to find essentially all the common variants, the variants that were present in 1% frequency or above in the population, with, if possible, a deeper emphasis on the coding regions. And not only to find them, but also to identify which individuals in the sequenced samples carried them. We eventually homed in on 2,500 people, so that would provide a sort of framework that was getting away from a single reference into which we could place new individuals. And that allows essentially extending the HapMap project, which was an earlier project based on genotyping. So the pilot project for the 1,000 genomes initially studied 179, it was 180, but we lost one of the samples essentially, from European ancestry, Yorubans from Ibadan in Nigeria, and from Beijing and Tokyo. Phase 1, which was completed, the sequencing was completed in 2010-11. The data analysis was completed last year, and this is currently submitted as a manuscript. Studies 1,100 samples about. We added populations in from the Americas and further populations from Europe, Africa, and East Asia. And we've now selected 2,500 samples on a plan basically of 5 continental groups. And then within each continental group, there are typically 5 populations of size 100 each. Slightly more complex in the Americas. And so we've added South Asian samples. And for each individual, the aim is to sequence genome-wide at 4x coverage with a whole genome shotgun, also deep exome sequence, and obtain genotyping data. Okay. So just to briefly tell you where we've got to in terms of data progress. The pilot project, so when we started, dbSNP, which was the catalog at NCBI of genetic variation, essentially, had about 10 million SNPs in it, variants, single nucleotide variants, not all of which were, some of which were false positives. The pilot increased that to 15 million, or discovered 15 million. But when we scaled up, there's an intermediate freeze with 600 samples. There were 26 million. And the phase 1 data set has nearly 40 million single nucleotide variants, to which are added about 1.5 million indels and 15,000 large deletions. As we've added more samples, by sharing information across the samples, we first increased our sensitivity, the degree to which we find variants known to be previously polymorphic. We've decreased the false positive rate, and we've increased the genotype accuracy. That's the accuracy at which we can say for any one individual whether they carry a variant. And I'll come on to show you that a little bit more about that on the next slide. And so now we've produced a sort of integrated picture for each individual of their 2 chromosomes and of which variants they carry on those chromosomes. So, people often ask, why do you only sequence at 4-fold coverage? On average, it's a stochastic thing, each individual. That could mean that at some places in the genome there'll be no reads. Each person has 2 copies of the genome. If you just have 4 copies, maybe they'll all come by chance, sometimes from one chromosome and not the other. So how can you possibly get an accurate sequence? And the reason is that we all share common ancestors, and so we share sections of sequence with each other at different distances. But as you increase the number of samples, you can identify relatively close relatives amongst the samples. So individuals who at this point in the genome share a common ancestor, and then share information between those. And that increases accuracy of detection of variants and of genotyping. So if we look here, the blue line shows the accuracy from the whole genome from the low coverage data. And this is the number of times that a variant is seen in the sample. So we've got a sample of about 1,000. So something that's present, and there are 2 chromosomes per individual. So something present at 1% frequency is present 20 times in 2,000. And you can see that almost all of those things are found here. In fact, if we drop down to things that are seen just 5 times in the sample, still 95% of those have been found. And things that have been seen just twice in the sample, though, it is true that something that's present only once or twice in the sample, we then can't share information so well because there are not so many other individuals carrying that variant. For a singleton, there's no other individuals, and you just have to rely on the data from an individual, and then we only have about a 30% chance of calling a variant. So that's the power to detect from the whole genome. It's interesting to look at the exome data. We still, in fact, lose power for rare variants in the exome, and that's partly because we're trying to control the false positive rate. We're aiming for a false positive rate of under 5% on the overall call set. And so we have to be extremely careful about variant calls. And when you get to very large numbers, coverage is variable, and it's easy to miss variants. So that's detection of a variant. What about genotyping accuracy? Well, here, if you look at the raw data, look at individuals without sharing, then the accuracy, the genotyping accuracy, flattens out at about below 90%. So we can't call accurately for each individual their genotype. But if we allow ourselves to share with using linkage disequilibrium, or which is what LD stands for, across samples, then again at 1% frequency we can get over 90% genotyping accuracy. And this is by a correlation measure to a gold standard or a standard genotyping chip. It's very hard to get gold standards or absolute truth. Here the exomes are clearly, they've got much deeper data in the coding sequence, and so therefore are more accurate again. Okay. So this is saying we've done pretty well. It seems by our original standards we've surpassed it. But it's slightly misleading because here we're looking at 1% frequency in the sample of people we've sequenced. What about a new sample of a new individual or a new set of individuals? So we've got external data which we can compare to the 1,000 genomes. And a good example is from this UK 10K project that Burkhard mentioned. This is a project to look at 4,000 individuals from the UK, or this component of the UK 10K, for all of whom we have phenotype data. We have a rich set of phenotype data. And we're sequencing them also at 6.5x depth. And so we can say something that's at 1% frequency in 2,500 samples. So this is an interim release from that project of 2,500 samples. Will be present about 50 times. So something that's at 1% frequency will be present 50 times in UK 10K. We get a good sense of what those are. And we're finding over 95% of those in the 1,000 genomes data set. That's the blue line in the... And actually, if we look at the part of the genome that's most easy to call, that's most unique, then that goes up to over 98%. Okay, so the other interesting thing from this graph is that if you look down here, things that are present at 0.1%. So sometimes people think the 1,000 Genomes Project is targeted at finding things at 1% frequency and above. But actually, half of the 0.1% population variants are also found by the 1,000 Genomes Project. So really we're finding, and the vast majority of variation found by the 1,000 Genomes Project is in fact rarer than 1%. It's in that area below. So we've got, there's a vast wealth of genetic variation we're finding certainly down to the 0.1% level and below, in fact. So that all looks good, but there are certainly outstanding problems. So for those, many people in the audience who are involved in this, it's quite technically challenging to call variants in sequence. And fundamentally, the reason is that the sequencing data isn't an end-to-end sequence of the chromosome. It's shotgun, random shotgun. We're getting short reads of 100 bases or so, some in read pairs. Some technologies give a few hundred bases. But they come from all over the genome. And so the strategy we use, as you're probably aware, is to map those to the reference. Read pairs help in repetitive regions, but there are parts of the genome where mapping is difficult. It also has to be, any algorithms we have to be fast. We've got, many terabytes of data in this data set. So, in fact, the figures here are for SNPs, for single nucleotide variants. As you can start considering variants which are in, so you can tell by the difference between these two lines, variants that are in difficult to sequence parts of the genome or repetitive parts of the genome, then performance degrades and coverage degrades. But also as you start to consider genetic variants which are more different from the reference than just a single point change, a deletion or an insertion or an inversion, some breakpoint, each of those progressively becomes harder to identify by a mapping strategy. Just to give some sense, people's genome sequences are nearly the same as each other. This is, in fact, the Venter and Watson versions of the sequence at one almost random position. There's about 1,000 bases on the screen. We've got about one difference in 1,000. If you want a 1% false positive rate, then that would be a 10 to the minus 5 error rate. But if you've got 1,000 individuals, you need a 10 to the minus 8 error rate per individual per base. And that's 1 in 100 million is a stringent requirement, and the more data one collects, the more stringent one has to be in combining information. So what are the sources of errors, and are we fundamentally going about things the right way? And I think probably not. We've reached a point where we're saturating on what we can do by mapping and variant calling. It's still creating problems, and we're missing things that people know are functionally important. So the sources of error, basically three. There are base call errors, and these come from the sequencing instrument, and to some extent the way the DNA has been handled. There can be mutations taking place before you sequence. But these are mostly independent, at least for standard technologies, not entirely, and we can model them. They're quite simple, and we can characterize them and then handle them statistically. There's also sampling error, especially with low coverage sequencing. We're only looking, it's a random process, and so we get sampling errors. But that's really very easy to handle statistically. And we can control it by read depth, and we can use these tricks of sharing data between samples. So that's manageable. Both of these are manageable. But the bigger problem is read placement errors and mapping. And so basically, it's knowing where this read comes from in the genome. And the way we do it at the moment is that we look for the best match, and we say, well, we'll trust that. Or you decide to what extent you think you can trust it. And the issues there can be errors in local alignment around indels. These are issues which we knew caused problems 20 years ago in BLAST and Smith-Waterman alignment. There are issues of assignment to the wrong copy of a known repeat. So it could be that we know the genome has multiple copies of some sequence. You get a read and you think, oh, it probably comes from this one. It looks most like this. But actually, maybe this individual is different, and it's actually coming from this copy. And the third and almost worst problem is where there's some piece of sequence the individual has which isn't really present in the reference at all. So we can't even know about considering that as an option. And in that case, what happens is we tend to plonk it down where it fits best, and that can lead to significant errors. So mapping errors are highly dependent. An individual's genome is obviously consistently different from the reference. So if something is mapped in the wrong place, if one read is mapped in the wrong place, then other reads will also be mapped in the wrong place from the same. And they cause almost all the real problems. So here's an example from Eric Banks from the Broad of where there are, in the original alignments, which were fairly sophisticated, there were a set of single-point variants called, apparently. And you can see, though, that all the reads in one direction show one sort of variant, and all the reads in the other direction show another sort of variant, and that's a sort of slight giveaway. And in fact, these can be explained by a large indel at one place. Because if you look closely, which only those at the front can do, the sequence along here is quite repetitive. And there's an extra copy of the repeat that can explain all of those single-point variants, including small indels with a single big indel. So for phase one, we came up with ways of, for the pilot, we sort of found these things, and then what we did is we said, oh, if there's too many variants all close together, we'll just not trust ourselves and mask them out. So that was a bit crude. For phase one, the next step that we're just publishing, we have a measure of local alignment confidence. In the Smith-Waterman dynamic programming, we can assess what the probability is, how confident we are in the alignment, in the sort of way that those who know our book may be familiar with. It's a posterior probability of the alignment. And we can downweight our confidence based on that, and that's what Li Hung did in his back approach. Nowadays, what we're beginning to do, though, is reassemble, look for new alignments around here. There are also errors due to mapping problems. Here's an interesting example. We discovered, Adam Orton noticed in an early draft set of calls that mostly the European, or the calls from the CEU population match those from the GBR, British population, except there are a few calls here seen in the British population, not seen in the CEU. If you went and looked where those were on the genome, they weren't randomly distributed. They stacked up right near the centromere, near the telomere, and in a few other places. And those are all known to be repetitive. They're full of satellite, horrible DNA. Basically, they're caused by mapping problems. So we've dealt with those. Those accounted for 1% or so of the calls. But essentially, the problem here is that the reference was incomplete. So the correct solution is to change the reference. The correct reference for each sample is unknown. And what we're doing when we're trying to map currently to the standard GRC H37 reference is we're mapping to the wrong reference. So what are the alternatives? Well, we could try and map to all known human genome sequence, but first we don't have tools for that, and secondly, we don't really know how to do that. We do have a lot of information, though, and really what we need to do, in my view, is use assembly-based approaches rather than mapping-based approaches. And assembly and mapping and search are all related problems. Also, of course, if we use assembly, then in a situation like this, where there's an insertion, we can potentially infer the entire sequence that's missing, which we could never do by mapping. So how do we map and assemble? Well, the central operation is string matching. And that gets into sort of very basic things in computational bioinformatics. And when you map, you want to find the best match and estimate how much better that is than other possibilities. When you assemble, you want to compare reads pairwise and build a consensus from overlaps. So there have been a lot of different approaches, hash-based approaches, and so on. But in the last few years, people have begun to use the Burrows-Wheeler transform FM index strategy, which is based on suffix arrays. And I want to spend a little time on that because I think it's really a beautiful data structure that captures the repeat information in sequences, and so captures the essence of what you want to do when you're aligning sequence. So traditionally, and in particular, there's a close connection with assembly. So traditional capillary sequence assemblers, from those of you who are old enough to remember them, before the days of new text sequencing, were heuristic and were based on overlaps between reads. There were heuristic methods, the first few from Roger Staden and Phil Green's FRAP. And then Gene Myers and Mihai Pop and others introduced graph-based methods, which were more kind of deferred decisions where there were conflicts in the data. But in general, these were order n squared. You had to look for overlaps between reads. And so they were fairly costly in terms of the numbers of reads involved. So Pavel Pevsner introduced De Bruijn-based methods based on k-mers and extending by one base at a time. And his Euler method was very influential, followed by Velvet, Abyss, Allpaths, and SOAP. And most of the current new tech sequencing assemblers are based on these methods. But they have a problem that the repeats that are longer than the k-mer require some sort of post-processing to thread to sort out. So they don't really use all the information in the reads in a clean way. It has to be subsequent steps. So if we wanted to go back to these overlap graph approaches, and Myers' string graph was a kind of very nice formulation, but the way that he did it, he found all the overlaps, and then he got rid of the redundant ones. So he found, let's say, an example here where these three reads overlap. He said, well, 1 overlaps 2 and 2 overlaps 3, and that's sort of good enough. You can then delete the edge that connects 1 to 3 because it's transitive and you can remove it. But he had to have a search process for identifying and removing those, and that was time-consuming for very large read numbers. So Jared Simpson, a graduate student with me, was working with these FM index structures and realized that, in fact, based on the FM index, you could find only the non-transitive overlaps. You never needed to go ahead and find all the pairwise overlaps and then remove the transitive ones. There was a process to find essentially the biggest overlap first, and then stop searching. So in fact linear time, you can find all the non-transitive overlaps and that immediately allowed us to start writing overlap-based methods for short read assembly. So it turns out that the FM index, which was introduced for search originally, is closely related to an assembly graph. It's almost the same thing. It's a sort of reformulation of it. It encodes all one-base extensions of a k-mer as a De Bruijn thing. It also encodes all these minimal overlaps, and you can compute the structure of the string graph in order n time. And you can also derive all De Bruijn graphs for any k up to the read length from it. So it encapsulates the whole lot. So Jared Simpson, Jared developed this SGA assembler, string graph assembler. It also uses the compression property. So another interesting property is that the Burrows-Wheeler transform, which we use now for search in Bowtie and SOAP and BWA, was originally introduced as a string compression method. And the compressed version of the index is both smaller and it's faster to operate on. So SGA for 40x human assembly, which is 128 gigabases of sequence, can assemble that in 54 gigs of memory, and 6 days, which is comparable to the time that SOAP and Allpaths take. SGA in the assemblathon last year came in the top 3, was third behind Allpaths and SOAP. Close to them. It didn't have the longest contig N50s, but it had the fewest substitutions, so it was highly accurate. partly because of, I think, of the overlap-based methods, and the second fewest misassembly errors. So that's all very well for assembly, but I started talking about genetic variation. Let's get back to variation. Can we use these assembly methods to detect variation? Well, yes, we can, and we can do it in a way that's reference-free, and that's, I would argue, a desirable thing to do. So a good example is if you want to look at de novo mutations by comparing a child to the parents. At the moment, what people do is map all the reads of the parents to a reference and the child to the reference. You can think of, but there's about 1,000 times as many differences between each of those individuals in the reference as there is between each other. So it's like measuring the distance between here and here by measuring distance to the end of the room and back again and subtracting those numbers. It doesn't seem a very smart idea. What you'd like to do is just compare the read sets for the children with those for the parent directly. Similarly, for somatic cancer mutations, comparing a tumor to normal. For population variation, just looking at the read sets for the individuals and ignoring the reference. And we can, in fact, look at individual variation by comparing to a reference. That's a perfectly legitimate approach, a synthetic read set. So we could do this by assembling each one first and comparing the contigs, but actually that's kind of time-consuming and misses the point. We really just want to look at the differences. So here's how we do it. We find unique k-mers present in one sample but not another sample, and then we locally construct the string graph around that. So we can extend outwards until we hit something that's shared, and then we can go back and fill in and produce the alternate haplotypes. And the complexity depends upon the number of variants, not on the scale of the problem. So here's this particular case, and there's a single point variant. Here's this applied to two yeast strains. You get a sort of set of shared nodes and nodes that are population strain specific. In this case, there's a single point variant, but the next one, which is just as simple as far as the graph is concerned, is a large insertion. So the method naturally finds insertions and deletions with the same ease as point mutations. So what about really applying it to variant calling? Well, first we looked at de novo calling, so comparing this individual, who, for those who know, is the CEU child of a trio that's been very intensively studied, to its parent read set. So this is each sequence deep. And we found 93% of the SNP calls that had been found and validated in the Conrad et al. Paper that was a big consortium based on the 1,000 genomes pilot data. We found extra calls, but they also had found 2,000 extra calls, which they then rejected through a whole validation process. So our number of extra calls is smaller than that. And we're going through validation both for these and also for indels. And they started to try to call indels, but they actually gave up. And so because of false positive issues on indels, we believe that there are clear, accurate new indels in there. This is a fairly manageable number to validate, and that's going on. We do lose power. I mean, I said we found 93% of what they found. These are graphs with, depending upon k-mer, for the overlap-based approach and the De Bruijn approach. You can see we don't find everything. That's on simulated data, and this is on the NA12878 data comparing to the reference. So we're only finding about 80% of all the calls in that for 30x depth. We've also used this approach on calls across a set of samples, and that's kind of interesting because people normally think for assembly that you need to have high depth. But again here, what we're looking for is k-mers or sets of sequence that are present in some samples but not other samples, so we can share across samples. And this is looking at 160 samples from African populations or populations African American. What we're doing is looking for a 61-base non-reference k-mer seen in over 4 times on both strands. Then we extend that out to haplotypes, map to the reference, and then we can use a Bayesian caller that we'd introduced earlier to make a final decision about accuracy. So this again worked in fairly reasonable memory. We called on chromosome 20, 220,000 SNPs, 38,000 indels. It's kind of interesting because this is a larger number than is often found in normal mapping. I'll come back to that. They look good by standard metrics of performance, quite high accuracy. We also found about over 2,000 multinucleotide polymorphisms where several different bases in a row had changed. And those tend to be dropped or ignored by standard methods, but they're 1% of all the SNP values, and we know that some of these are functional. If we look at the Indel size distribution, we're calling variants right out to 600 to hundreds of bases on either side. Again, typically read mapping approaches, well, you have to start using different strategies to go beyond 10 bases or so, or 20 bases. There's a slight bias towards deletions, but that's actually expected in a population sample for reasons I could go into for those who are interested afterwards. So here's an example of a 13-base deletion and a 4-base multinucleotide polymorphism. And this is interesting because there are only 3 reads present in the original BAM, but there's 43 reads overall in the assembly. So the vast majority of the reads that correspond to this have been missed by the original alignment, and any local assembler would have missed them. Okay. So overall, I think there are methods available to start thinking about, although we've got a long way with a reference sequence, it's been important, and mapping-based approaches, that ultimately the data that we get from an experiment is a read, and from many different types of experiments are sets of reads, and we should try and develop methods which work directly from the sets of reads to make inferences about the things that we're interested in, rather than having to go through a mapping approach. We should bring those methods in, the scope for doing that. And I think the scope for doing this beyond just finding variation, but also for asking population genetic questions and potentially association questions. So what are the pros? There's no bias due to the reference. All samples are equal. And we can find more divergent mutations. It is true we get less long-range continuity. We need to place the contigs. The reference is important. It provides a coordinate system. I don't want to give up on all that knowledge. And clearly, any assembly-based approach can't put things together from one end of the genome to the other. And also, we do miss something. The read pair-based mapping methods are pretty sophisticated, and currently the assembly methods can't access all the genome that they can access. Some of these things will be addressed by longer reads, which we hope are coming. The PacBio reads, as we heard this morning, are in the first keynote, are attractive, but they're currently too expensive. And also there are hierarchical partitioning approaches where, like Jay Shandor has proposed, where one takes pools of intermediate scale clones like phosmids to sort of break up the genome into smaller pieces and allow it to phase and assemble hierarchically. Okay, so I want to change and use the sort of second part of my talk to go beyond just finding the genetic variants, although that's important and it's the sort of foundation to really get a good picture of genetic variation, to think a bit about association mapping. And those of us who were here in the session earlier heard from Ed Marcotte about trying to dig into association studies, the results of them. But typically in humans, association studies at the moment operate between genetic variants and whole organism phenotypes, disease, or these things are important medically. A lot of the funding comes from medicine. But we know that in biology, the mutation here doesn't directly cause a disease. There's a cellular effect initially on a transcript or a protein product. That'll lead to some sort of organ or tissue effect, and then those combine together to a disease. And we know now from genetic association studies of disease that often there are many variants with weak effects at this level, but often at this level the effects are strong. And this is a slide from Manolis Demitsakis' review a few years ago. So many people have talked about expression, have done expression QTL, eQTL studies. I think there have been lots of talks in the meeting about them. And often we can start filling in this picture. So the eQTLs are basically phenotypes at this level, and sometimes they are directly involved in the effect of this variant on a disease is via the expression of a gene. Some signals, they're often much more strong signals. They can be visible with just 60 samples rather than hundreds or rather than thousands of samples for disease association. You can find hundreds to thousands of cis eQTLs just from small number, or once you get up to hundreds of samples. Trans eQTLs are where a variant one place in the genome affects expression somewhere else in the genome. And there's a whole set of statistical computational methods for processing those and extracting further information. But I think we should be going beyond mRNA expression as a phenotype, and it's been underused, the approaches of using human genetics to study cell biology and basic human biology. And actually, I think some of the greatest strength of these new association genetics are going to be in understanding basic biology, and that helping feed, as it always has done, into medical research. So we first looked at small RNAs, and we sequenced small RNA from 131 fat samples from the set of Twins UK subjects. So this is a cohort of twins that had been studied, had already got a lot of mRNA data in the mother consortium. We got on average 2.3 million reads per sample, and we found evidence for about 400 microRNAs and 170 SNO RNAs. These are small nucleolar RNAs involved in nucleolar function. But basically, most of the data came from microRNAs. And these were the ones with more than 1,000 reads. There were some other fragments of other sorts of RNA that we found as well, but didn't have enough read depth to start to look at them genetically. And when we looked for QTLs, there's within 100 kilobases of the transcript, we found 14 significant cis-QTLs in this at a 5% discovery rate. So these were significant at a genome-wide, or no, at FDR. And 14 might seem small compared to eQTL, standard mRNA eQTL studies, but actually, if you think that we're only really looking, there's a smaller number of microRNAs to start with, actually the ratio is about what you'd expect, and it's comparable for the number of mRNA QTLs you see in 150 samples. So let's look a bit more at those. 8 of those 14 turn out to also be eQTLs for messenger RNA. And those are messages coming from genes near the microRNA, but they're not always the closest gene, and it's not always the same sign. So there's some variants where a genetic effect, a genotype is affecting the expression of a microRNA and a protein coding gene, which is some distance away. So these possibly are in enhancers. It indicates the complexity of gene structure. Now you might know that often microRNAs are transcribed from or they're cut out of the introns of messages. So you might say, well, that's what's going on. These are microRNAs coming from the same transcript as the mRNA. But interestingly, that wasn't the case, although microRNAs from a cluster were correlated. So there was a surprising lack of correlation of expression between microRNAs which came from the transcript of an mRNA. So there's clearly a separate regulation of the production level. We can ask, are these microRNAs on the way to some phenotype? And we're looking in fat tissue, and 4 of them are nominally significant at 0.05 level in a BMI genome-wide association study. So that's not enough that you would have picked them out from scratch, but it is significant enrichment below 1%, binomial p below 1%. If you look more generally at whether there are microRNAs which are associated with BMI in the sample, there are 41 of them at that level. But those ones, that's not so meaningful. These ones here, the genetic variant is associated with the microRNA and with the phenotype. If you're just looking at correlations between expression level of the microRNA and the phenotype, those could be, we don't know the direction of causality. It could be that, and we know that this is true, that higher BMI, higher obesity can change the nature, the state of tissue, and change the nature of gene expression. Okay. So what about microRNAs and targets? And actually, this is why I started the experiment. And I thought, what we'll do is we'll find microRNA variation and genetic variation and mRNA variation. And the microRNAs we know affect mRNAs by controlling their transcript abundance, and I thought I'd find correlations between microRNA expression levels and their target mRNA levels. So this was a bust. Like all good science, some things seem a good idea and don't work out, and it's quite interesting. It turns out that there's no evidence for negative correlation. There's no real evidence between microRNA and mRNA levels. So we took 522 validated miRNA message pairs, target pairs. So these are which have been experimentally validated. And there's a sort of pathetic correlation. We also took a whole big set of predicted pairs. So we all know, I mean, those of you who work in this field know that prediction isn't great, but it's not hopeless. And there's 200,000 predicted pairs, and we thought that there would be some sort of signal in that, but that's an even more pathetic correlation. And furthermore, there's no significant, if we take the most significant, of course there's a scatter, the most significant set of the most negatively correlated pairs, then we can't see any evidence for seeds of the microRNAs. So we really don't see any evidence for anti-correlation of microRNA and mRNA expression levels, which is kind of interesting. So where does that leave us? Well, microRNAs are regulated transcriptionally, at least in part via enhancers, which they share with protein mRNAs in non-trivial ways. They must also be regulated post-transcriptionally, since they don't typically correlate with co-transcribed messages. They do contribute to end phenotypes, but their lack of strong correlation to target mRNAs supports the proposal some people have made that the primary role of microRNAs is in buffering mRNA levels, rather than actually switching them, using a microRNA as a switch. And that would then be consistent with this observation made on this page. So that's anyway at least consistent. I don't know if I would say it proves it. It certainly doesn't. So now I've got a little time left. So we want to go, you could say it's another expression phenotype, a microRNA. We clearly can put those in and start to learn some things and get specific and general information. We can now also use sequencing, as people know, for ChIP-seq and for all sorts of assays. So we started asking, can we do ChIP-seq QTLs? We started with CTCF. It's a binding factor involved in demarcating regulatory domains. It's sort of known as an insulator protein. It's a good choice. It's a strong motif, robust ChIP-seq antibody. It's been used in ENCODE as a kind of reference core pull-down reagent or data set. And there are many binding sites throughout the genome. It's sort of fairly pervasive. So we did ChIP-seq of CTCF in 60 cell lines from the 1,000 Genomes pilot. These are lymphoblastoid cell lines. 40 million reads per sample. It's a collaboration with Vishy Iyer, Greg Crawford, Jason Lieb, and Ewan Burney. Here's some data showing you these are locations of CTCF binding sites, and you can see variation at these and different levels of, and there's some controls down at the bottom and somewhere or other. And we get strong QTL signals, even from 60 lines. The graph on the left shows, as we do various sorts of data processing, how the number of positive hits varies with FDR. The right-hand side is a QQ plot for those who know such things. This is the expected p-value and the observed p-value. A control, if we permute the binding regions then we get essentially flat line, non-inflated, clean line, but there's a strong divergence from that, although you can see that we're actually not inflated down here, which is what you want to see. So we find 1,100 binding regions with significant QTLs at 1% FDR out of 107,000 in a 100 kb window around the binding region. And on average, there's about 30 SNPs per binding region. So we have full sequence, we can see they're correlated. That doesn't mean that there are 30 functional associated SNPs, because there's a linkage disequilibrium. And so, in fact, in most cases, if you find the best SNP, then all the others are correlated to it. And there's only a handful with independent signals. So we know what the binding motif is, and we can go and find the binding motif in the binding region. And then for nearly 800 of the binding regions that we pick up as peaks, we can find a strong motif match. 224 of those have a QTL within the motif. That's of the... So you can see here, and you can see there's a sort of exquisite matching. This is the degree of conservation in the motif lined up. And you can see that where the motif is strongly conserved, then there are a lot of strong effect QTLs. And where it's not so conserved, there aren't so many. So this all looks good. But it's interesting that it's only a quarter of the QTLs can be explained by interfering with the binding motif. So where are the others? There's new biology out there. There are variants which are affecting CDCF binding, but not interfering with the binding motif. And we know that because we have full sequence, basically. So we're now going with MEME and these sorts of things to try and pull out new things. So what's the future? I think there's a very strong future for cellular association genetics. And I think we've been hearing a lot about networks. And that's a key strength of this meeting, people are interested in this meeting. But by adding in genetics, genetic association has a very nice Mendelian randomization properties and also gives us causality. It's not true that the expression or some cell biology changes the genome sequence. So you can really infer causality in a strong way with genetic association. And that's been powerful repeatedly in biology. We can do all sorts of other assays, DNase 1, there's papers about this coming out. But I think it's going to be important to put together results. So far, most of these studies have been done in lymphoblastoid cell lines, which are these EBV transformed cell lines, or primary tissue. But the problem with primary tissue is it's got finite access and it's unclean. So to finish with, I want to tell you briefly about a new initiative that we are starting up. It's a network to make and bank a panel of induced pluripotent stem cell lines from over 500 normal people and over 500 people with monogenic disease. It's recently been funded in the UK by the Wellcome Trust and the MRC. There's a whole set of partners, so it's a collaboration with the Sanger Institute, Ludwig Vallier, who's an IPS biologist, Daniel Gaffney, was with Jonathan Pritchard, King's College London, Fiona Watt is a cell biologist who works on stem cell biology and signaling pathways. And so we're going to have signaling assays and also proteomic assays. And there are clinical collaborators providing the clinical samples. And you and Bernie, certainly not least at the EBI, involved in data analysis and making the data set available. So the aim is to make the data and the cell lines available, and there'll be fully open access for the normals in the same way that the HapMap and lines are. And I hope that we can really integrate information on a core genetic resource. So there's a set of slides, and I think I'm running out of time. So on pilot experiments with this. So there's been some skepticism in the stem cell community about the feasibility of doing genetics in IPS lines. One argument they make is that there's lots of mutations caused by the IPS reprogramming experiment. So here we have Kosuke Yusa, who's a postdoc with Alan Bradley and Ludwig Vallier, started all from one individual. This is, can't read it, fibroblast and blood sequencing. These are EPCs, endothelial precursor cells, which have the property that you can make APS lines from them. They're isolated from blood, and you can grow them up clonally, so you can clone them out. So this is a somatic cell that you can grow clonally. There were 12 mutations found in an exome in that particular EPC line. From that line, we made 5 IPS lines. Each of those added 1 to 5 new mutations. In fact, 2 of them turned out to be the same. So we re-isolated the same thing. So there's 1, 3, 4, and 5 mutations. There are typically 15,000 exome mutations between individuals. So the scale at which one is adding mutations by IPS reprogramming is very small. Of course, it could be true that one of those is functionally critical, but it's, in my view, one can handle that degree of variation. It's not a major effect. This is showing if you have one person, IPS lines from one person, isolated from different tissues, from fibroblasts, keratinocytes, or EPS lines, they cluster together and they're indistinguishable, and they don't actually cluster by tissue of origin. So this is RNA-seq data. These cluster with the ES cells, but in fact the IPS lines have more similar expression patterns than different keratinocytes from the same person or different fibroblast isolations from the same person. So they behave in a clean fashion. Here we're looking at, again, it's RNA-seq data. This is a known gene which has in LCLs. This top panel is from HapMap LCLs. There are eQTLs at nearby SNPs. So these are SNPs that decrease or increase expression. And And here we've got allele-specific measurements from RNA-seq data from our IPS lines, and we can see the same signals in the same direction. So we can see genetic effects in these lines, so I think there are good prospects of that. And finally, and I think quite excitingly, those people who've worked with induced pluripotent stem cells, which are cells that come from different tissues in the body, but you reprogram them back to look like embryonic stem cells so that they're pluripotent, and then you can start differentiating them down different pathways. Different cell lines have different abilities to differentiate. There are these protocols for differentiating them, say, towards endoderm. And some cell lines, it's very easy. They just kind of go there. And others, it's a lot of work, and only a fraction of the cells go there. And that's, of course, in the body, they all come from somebody who had endoderm and ectoderm and mesoderm, so it must be possible. But, of course, we're working in a tissue culture situation and not fully understanding the process, so I think it's not so surprising. So the observation here is we've got 39 lines from 14 individuals, and they behave systematically between individuals. So this individual, there's 5 lines, they're all hard to get to express endoderm markers. So the same methodology has been used on every line. These are controls. These are 2 lines that convert well. These are 2 control lines that weren't driven towards endoderm expression. This individual, they differentiate well. This one is poor. This one is well. There's some scatter, obviously, but there's clear signal of correlation with the individual rather than with a particular line. So, in summary, I think we've got potential not only for sort of proximal cell biological proximal functions, but also for starting to look at the genetics of differentiation and of cell function. And I'm quite excited about starting to work with cell biologists who measure signaling pathway activations. I'm sure we'll find variation in those processes, and we'll start to be able to tie together the networks involved. So just to finish and acknowledge contributions of people in the group. So Jared Simpson and Case worked on the assembly and variation. These other people here have worked in various ways on collecting DNA and genetic analyses in the group. 1,000 Genomes Project, of course, has been a great consortium and collaboration, and there have been many really wonderful people, actually. And working with public data sets in the open, I find, has been extremely stimulating and has been generated methods and data that other people have used in all sorts of interesting ways. The microRNA QTL study was carried out by Leo Parts together with Asa Hedman and Cecilia Lindgren at Oxford, using data from the Mother Consortium. And the CTCF QTL experiment, Zhihao Ding in my group has led that, but there are many other people, particularly in Vichy Ayers and Ewan Burney's labs, who've been key to that. So thanks very much.
Questions?
Richard, right here on your left. Hi. Great work, obviously. I have a question about the chip QTLs for CTCF. Namely, how often do you actually not find a SNP in CTCF per se? And do you actually find a SNP in a neighboring motif? And have you tried to sort of explain away the ones that don't have an obvious SNP in the motif site? And also looking at your actual figure, the axis was perhaps a little off, and I'm just wondering if the additional peaks are not due to CTCF.
This one?
Yes.
They do line up. If they didn't line up, that's because I didn't line them up. So it's not true that they're always nearby. So you can look at where the nearest SNP is with any signal, QTL signal, to the motif. And they're peaked around the motif, but there are ones that are kilobases away. So, and in fact, there are ones that are kilobases away from the whole binding region. So it's clearly true that there are genetic variation in binding of CTCF caused by variants outside the binding region entirely. We're basically trying to go down the route of correlating to other sorts of annotation and other sorts of other binding motifs. That's work in progress. It's a kind of work in progress.
So even if
slide.
we don't identify the motif, have you looked for, say, conservation as a signal? Is there a signal there?
That's a good question. No, that's a clearly good question. Of course, often, there's often tens of variants, so you have to be a little careful statistically, because it may not be the very top one, it may be another one that's causative.
Thank you.
Hi, Richard. First, I want to know that I'm a kind of community member that use your population dynamic data in the two yeast species. So for that, thank you. But my experience in using the population dynamic data is that, particularly referring to my experience with the yeast population genome sequence, is they have very low coverage, and thus many genes were not complete. The sequences, particularly some of the most interesting genes in yeast genome. So, I'm glad that you are going to sequence human genome with higher coverage, but I was just wondering if by any chance you have considered to resequence the yeast population genomes with higher coverage.
So you're referring to some data we collected over 5 years ago now. In fact, before new text sequencing, with capillary sequencing, we sequenced a set of 70 yeast strains at low coverage. And that was a sort of demonstrator project to me to show that one can find variation and let in. But a lot of technology has moved on since then. So certainly if I started again with yeast, it would look different from how that data set does now. And we're a victim of having produced an initial early draft of that and then moved on to human. It's, however, not very expensive. It's in fact a few tens of dollars to sequence yeast strains. Certainly.
So everybody can do it.
So I kind of think it's out there for those who care about yeast population genetics to produce a state-of-the-art modern version of that. It's certainly feasible to do a better job. But I should say, as you get to smaller and smaller organisms, a higher fraction of their genome and of their genes are often held in repetitive, non-alignable, non-copy number, simple allelic copy number 1 sequence. A lot of functional genes in yeast are in subtelomeric regions that are not well represented in the reference, and there's serious strain variation. So the assembly-based methods, I think, are going to be all the more important for, and to some extent we did go into that in that paper. A lot of the things people are interested in require assembly-based methods, and you can't get at by alignment to the reference sequence. So in particular, in those organisms, that's going to be important. I have a sort of hypothesis that every organism has 5 megabases of, every species has 5 to 10 megabases of non-allelic copy number variable sequence.
All right, thank you.
Is there a way you can go back to the distribution of Indel length? Yeah. So can you give some insight about what happens to Indels of longer?
Well, there are not very many on this scale. I can't read it here. I don't know my glasses.
You just don't try to detect them?
Oh, no, we do.
They're not
Sorry.
visible?
We find them out to, of the order of a kilobase. It's just that for drawing the graph, I truncated at plus or minus 10. So there are fewer, though. I mean, the total contribution of them is, per indel is larger because it's more sequence, but overall, the contribution is greatest for small ones. So I think many people have seen this sort of pattern. Yeah.
So when you say indel here, now we don't have a reference genome, and I guess you're comparing one genome against the other.
Yeah.
So you should probably expect a symmetric distribution.
I see. Well, no, so this is complicated. This is actually put on the reference, but...
I mean, if it's against the reference, then I take my question back.
No. Yeah, I guess that's right. But actually, it turns out that directionally, there are more deletions than insertions on a small scale with time. So it's not a reversible Markov process. There's probably what's happening is there are duplication processes at a large scale and transposon insertion, and then there are deletions at a small scale. So rare events tend to be even against a reference which is randomly picked. As you look at a large sample, you'll get more deletions than insertions because you're seeing rare events.
Hello. So I have a question about the assembly method that you developed for the assembly method. Can you say a few words about the base call errors? How do you take them into account when you work? Base
call errors in the assembly. Yeah. So essentially, you have to clean up k-mers which you don't see sufficiently often, which don't have support from other k-mers. So, or where there are, and you can do it at the k-mer level, which is a standard thing to do. So most assemblers go through a read cleaning exercise up front, or a truncating exercise from the, often Illumina reads, or most sequencing processes, it's true. At the end of the read, the error count goes up. So the first thing you can do is just trim the ends of the reads based on read qualities. That's a sort of crude thing to do. Then you can look for k-mers that are not supported by any other read. But then you can get into trouble in very repetitive sequence where there's very high depth. So you can be slightly more sophisticated. There's in fact a whole literature on error cleaning from reads.
Okay, so using some standard.
So we used an error cleaner. There's a slide I took out actually, a k-mer-based error cleaner. But then we also have an overlap-based error cleaner, which is a bit more sensitive but more costly. So it's in the space of standard error cleaners for assembly.
Okay. Thanks again, Richard.